Assalamualaikum warahmatullahi taala wabarakatuh. I am Muhammad Zahruddin bin Sariman from Faculty of Plantation and Agrotechnology, University Technology Mara, UiTM, Cawangan, Melaka, Malaysia. So today I would like to present my study on title Effects of Various Photo Period Toward Lola Bayanda, Growth Using IoT-Based Lighting Control for Indoor Hydroponic Farming System. So my presentation outline, so we start from introduction and then objective and then follow up by materials and the method used in the study and then the data collection result in the study and then the data analysis and then finally conclusion and also the recommendation. So without any further, I would like to proceed to the introduction. So sustainable agriculture is actively developed to ensure food security, which is a major threat to the world population in 2050, as projected by the Food and Agriculture Organization. So due to the increasingly limited agricultural land, so nowadays it makes indoor farming is the best. So indoor farming is the method of growing crop in vertically stacked layers by the implementation of hydroponic and also the artificial light. So the use of light emitting diodes LED mimics the sunlight for the plant growth has been widely used. So according to the Ozonis et al. 2015, the plant productivity influenced by the light quantity and quality. So the light quality includes the intensity, which is the fluence rate, and also the duration, which is the photo period. Meanwhile, the light quantity is the wavelength composition, which also important for the plant growth and photomorphogenesis and tissue composition. So in each of the light spectrum affect the plant response has been discussed from many researchers. So for instance, red and blue light are most efficiently utilized for photosynthesis. Blue light influence stomatal opening, plant height, and chlorophyll biosynthesis, while far red light stimulate flowering in long day plants, and red-far red ratio regulates stem elongation and branching, leaf expansion, and reproduction. And then green light penetrate deeply in the leaf mesophyll layer and reach the lower and inner canopy level, promoting photosynthesis in the deepest chloroplast and in the less irradiated leaf and providing signal to respond to the environmental irradiance. Hence, it will improve crop productivity and yield. And also a detailed review on plant response to light spectrum of artificial lighting in vegetable and also ornamental crop in terms of growth and photomorphogenesis and the state of the art of the research on LED in the greenhouse horticulture has been discussed. However, the effects of the various photo period on the plant growth is still very few discussed. So therefore, my objective is to investigate the effects of different photo period LED lighting on morphological of Lola Bayonda to determine the best photo period treatment for cultivation of Lolo Bayonda. The research hypothesis was cultivation of Lolo Bayonda seedling under the effects of light with varying photo period would differently affect morphological properties. So the various exposure time of light will be measured as variable parameter, and the nutrient content, pH, and water level of the solution is a constant parameter. So the parameters are measured and controlled by using monitoring IoT-based system. Okay, next we go for material and method. So for material and methods, so we first is seed preparation for Lolo Bayonda. Okay, and then hydroponic growth chamber setup, photo period automation, and then design of experiment, DOE. So without any further, we go for the first method, seed preparation for Lolo Bayonda. The seed are pre-sown on a cubical sponge with a dimension of 25 millimeter each side for germination process. And then it is kept moist by using tap water for 15 days before transplanting them on the growth chamber. And then only from the 10 seed germinated, so only 6 of the best seedling are selected to be transplanted at the hydroponic system. So next, for the hydroponic growth chamber setup, so the hydroponic system was constructed by a hydroponic gutter filled with the hydroponic nutrient AB solution, which the initial pH of 6.35 and the electrical conductivity EC of 1.33 millisecond per centimeter. The hydroponics nutrient AB solution and pH level were taken weekly to maintain the consistency of nutrient supplied to each plant. So the pH water level in the system has been maintained around 5.5 to 7, and the EC level around 1.3 millisecond centimeter to 1.4 millisecond per centimeter. Use pH meter and EC meter to measure nutrient concentration manually. So this system use the nutrient film technique NFT to consistently deliver nutrient to the plant, requiring a 12-volt DC water pump. And then optimal control can be provided by continuously using the DC voltage for operating the water pump. And then bypass pipe was made to keep the water flowing slowly and consistently so that it can supply nutrient to the bare roots. And then the planting distance between the plant was 15 centimeter, and then the planting distance between the center of the hydroponic bed and the planting hole was 10 centimeter. So next is the photo period automation. So for the photo period automation, so we are using an ESP32 for a control growth like LED period for each treatment. And then the ESP32 has a Wi-Fi module connected with Wi-Fi to provide and monitor the data in the Blink platform by using the software on our phones. And then the data from the Blink platform was used a supercard widget to display the light data for each level fair treatment. And then the light source was used LED grow light, AC 240 volt, 240 volt for each treatment level with the red to white to blue ratio RWB is 3 to 1 to 1. So which the length of LED grow light is 50 centimeter. The LED grow light was placed on the center of the hydroponic bed, which 3 LED grow light for each treatment, except for the first treatment. And then for the design of experiment, so there are 4 treatments, namely T1, T2, T3, and T4, which represent the photo period of 0, 12, 18, and 24 hours daily. The responding variable of the experiment are the height, average leaf size width, average leaf size length, leaf number, root length, and the weight of the Lola Bayonda. And then there are 6 replications of the experiment for each treatment. So the planting period for each replication is within 30 days. So table 1 is the a table for data collection, okay, for my design of experiment. So next, so this is the result gathered from my studies. So through the observation, so the plant subjected to treatment 1 were observed to have died after 2 days after transplanting them. And then plant that subjected to treatment 2 is the value of the growth is maximum for the all of the parameters. So next, we proceed to the data analysis. Okay, for the data analysis, all the treatment were analyzed with single factor analysis of variance ANOVA to all the parameters tested to determine their significance difference. So wherever there are significant result, two keys B was applied to determine the pairwise comparison between all the treatment. So analysis was performed with IBM SPSS Statistics version 22. So the figure 3 and figure 4 is the values of means of plant height and also means of leaf width. So for the figure 3, so the means of plant height for treatment 2 is the highest, and then followed by treatment 3, treatment 4, and then the treatment 1 is the lowest. So and then for the means of leaf width, the value of treatment 2 is highest, and then the value of the treatment 1 is the lowest, and then the treatment 3 and treatment 4 okay, shows no significant difference. So next, we move on to the figure 5 and figure 6. So figure 5 is the means of the result of the means of leaf length. So the highest value still on the treatment 2, and then the lowest is the treatment 1, and then it seems goes as the figures 4, which is the treatment 3 and treatment 4 shows no significance of difference. And then for the figure 6, it means of the leaf number. So all the treatment shows the significant difference. The highest is on the treatment 2, and then the lowest is on the treatment 1. Okay, and then we go to the figure 7 and figures 8. So the figure 7 is the result of the means of root lengths. So all the treatment show the significant difference. So the highest on the treatment 2, 24 hours, and then the lowest still on the first treatment, T1. Okay, and then for the result of mean of weight, so treatment 3 and treatment 4 shows no significant of difference. So the result shows that there were a significant difference in all the parameters, which are the plant height, leaf width, leaf length, leaf numbers, and root length, and the weight between all the treatments mean. So it can be summarized that the treatment 2 was found to be the most significantly higher than the other treatment in all the parameters tested. However, treatment 3 and also treatment 4 were found not significantly different from each other in terms of the leaf width, leaf length, and the weight. And then the treatment 1 was found lower than the other the lowest of the other treatments in all the parameters tested. So as the conclusion, the duration of the light exposure to the lettuce Lolobayonda affected the growth of the lettuce Lolobayonda, such as height average, leaf size, leaf number, root length, and the weight. So based on the study, the long photo period of the Lolobayonda with the combining of the LED grow light with the red to white to blue ratio 3:1 increase the growth of the lettuce. The treatment to 24 hours photo period treatment was resulted in the highest growth compared to the others treatment, 18 and also 12 hours. However, the increase of the photo period has increased the electric power consumption, which increased the operating cost. Therefore, the photo period of 12 hours per day provided by LED RWB ratio 3:1 was recommended for hydroponic lettuce production under a controlled environment to the company that wants to save their cost. However, a photo period of 24 hours was recommended to the company that wants to boost their lettuce production. So this treatment decreased the cycle of the Lolobayonda and gain more profit than the photo period of 12 hours. So these are the sums of the reference that been used in this study. So that's all from me. Thank you. Assalamualaikum warahmatullahi taala wabarakatuh.